And I would like to introduce the second speaker, Dr. Lena Allweiss, for University Medical Center Hamburg-Eppendorf in Germany. She's going to talk about the RNA interference-mediated secondary mode of action and focus in particular on the impact that this type of therapy might have on the cccDNA. Thank you, Lena.
Thank you for the opportunity to talk at this meeting. And I was tasked to talk about the secondary mode of action, namely the impact on the cccDNA. So just to remind you quickly again, HBV RNAs are all transcribed from the cccDNA, and some can also get transcribed from integrations, in particular the HBS mRNAs. And RNAi can target all these different RNAs by putting a target here in the common 3' end, but because this region is often absent in integrations, the target has to be put a little bit upstream of DR2. And then both integration-derived and cccDNA-derived RNAs can get targeted. So this is the primary mode of action of RNAi. On one hand, it will lead to the degradation of the pregenomic RNA and with it the inhibition of HBV replication. And on the other hand, it will degrade all subgenomic RNAs from cccDNA and integration and abolish the production of HBV proteins. And in particular, the reduction of circulating viral antigens is promising the HBS because there's the hope that it might lead to HBS loss, function cure, and immune restoration. Because it has been known that chronic HBS exposure is linked to immune dysfunctions and the inability of viral clearance, as we've known for many years and also heard at this meeting again. So that's the primary mode of action. What is the secondary mode of action? The secondary mode of action comes with the degradation of the HBX RNA. And this regulatory protein, the HBX protein, can then hinder the production of transcription of all cccDNA-derived proteins and RNAs. And why? Because this regulatory protein is essential to maintain transcription from the cccDNA, as was shown in several preclinical models, and then this will hopefully lead to the silencing of cccDNA. So in the next 15 minutes, I would like to talk about in detail about this mechanism. I will also touch upon related questions and its relevance for the treatment of patients, of course. So unfortunately, there's very limited data in patients about this secondary mode of action. So I have to go back to preclinical models, and I will show you some data in HBV-infected humanized mice from our lab. So we treated HBV-infected mice with siRNA, which was LNP formulated for 4 weeks, and the target site was here in this region, so it would trigger degradation of all cccDNA-derived RNAs. And indeed, it worked. We saw a strong reduction of all serological viral markers and intrahepatic viral markers. The only viral marker that didn't change was the cccDNA, but that was expected because RNAi shouldn't interfere with established cccDNA. And very interestingly, we could also see that indeed the HBX protein was strongly decreased. So the first part of the mechanism worked. So what happens now? To understand this, I have to go back and explain the function of HBX. And I guess you all know that this is a regulatory viral protein. Many functions have been described in the last years, but it became clear that probably the main function is the degradation of the restriction factor, the SMC56 complex. And it does so by interacting with DDD1, which interacts with an E3 ubiquitin ligase, which leads to the ubiquitination and proteasomal degradation of this complex. And this complex is a host restriction factor that hinders cccDNA transcription in the absence of HBX. And just a few words about this complex. SMC56 is actually one of the 3 main chromosomal maintenance complexes in a cell. It's a 6-membered complex, and it's involved mainly in DNA repair during replication. And it was recently also shown that it can recognize unusual DNA structures, such as DNA supercoils, and then binds and compacts them in an ATP-dependent manner. So how does that look like in preclinical models? First, I'd like to show you some data from HBV-infected PHH. And here we can see that there's SMC56 protein. This is one member of the complex in uninfected cells, but when in cells are infected, we have HBX, and this complex is degraded. So there's an inverse correlation, and you can also appreciate this in this nice 3D reconstructions. What about in vivo? In our HBV-infected humanized mice, we actually see the same pattern. In uninfected mice, there's SMC6 in every nucleus, but when they are infected, which you can see here with the core staining, this complex is degraded. So now that we saw that this complex is degraded in a normal infection, of course, we wondered what would happen now that we have reduced HBx protein with the siRNA treatment. This was also investigated in cell culture. Here, siRNA treatment was used to degrade HBx, and with the reduction of positive cells, infected cells, there was an increase of SMAC6 positive cells. This is the percentage of positive cells. And of course, we analyzed this also in vivo in humanized mice. And here I show you RNA in situ hybridization for the viral RNAs, which are strongly produced in untreated mice. But then upon treatment, at end of treatment, we see that SMC56 protein actually comes back. So and what is also really interesting, I think, is that the siRNA seem to work in all the cells. So there's no cytoplasmic HBV RNA left. But only in some cells, like roughly half of the cells, we see that SMC56 comes back, and then the production, so the transcription, is completely blocked. We also checked with chip assays to see if this complex is actually associated with the cccDNA. We chipped for another component of this complex, NSE4, and we see there's a strong association of NSE4 and with it the complex on the cccDNA. And just as a side remark, we also treated mice with pegylated interferon in this study, and we actually see a very similar effect. So now we have a reduction of HBx and a transcriptional silencing of cccDNA through the comeback of SMC56. So the next question we wanted to address is how SMC56 mediates this transcriptional suppression. For this, we had a look at the epigenetic modifications of the cccDNA. And here, as an example, I'm just showing you one classical active histone PTM, post-translational modification, and one repressive one. And we saw that the active one is highly enriched on the cccDNA, and the active one, the repressive one, is absent. So this is in line with an active transcription, but surprisingly, when we treated the mice, we didn't see a change. So we believe that together with the other data I showed in the beginning, there might be a physical compaction and sterical hindrance of cccDNA transcription rather than a classical epigenetic suppression. I also want to show you data from two other recent publications that try to elucidate this mechanism in more detail. And both studies actually found that SMC56 entraps episomal DNA in an ATP-dependent manner, and then recruits these complexes to PML nuclear bodies with the help of another cofactor, SLC52. And in this study, they could actually show that HBV DNA was recruited to these PML bodies. Of course, there are still many open questions. We don't know exactly how the mechanism works. We don't understand exactly how episomal DNA is targeted specifically. So there's more work going on. But I think the burning question here is, of course, if we have the silencing, can it be maintained? Can it be one of the reasons, as John alluded to, for the sustained suppression in siRNA-treated patients that we do observe? So we wanted to analyze if cccDNA suppression is maintained post-treatment. For this, I have to go back to mouse data again. And we followed up these treated mice for another 4 weeks without any treatment. And in one group of mice, we also treated them with the entry inhibitor bulevirtide because we wanted to shield hepatocytes from new infections to be able to actually analyze the silent cccDNA in these hepatocytes. So when we did this, we saw, however, that all the mice rebounded. In the serum and in the liver, when we stopped treatment, we saw a rebound. So then we wondered what happened to the silent cccDNA molecules. We did again the chip assay, and after follow-up in the untreated mice, we saw that the cccDNA association with SMC56 was lost. But when we blocked entry, we saw that SMC56 was still associated with the cccDNA. And in this double staining assay, we also saw that during follow-up, when entry was blocked, we saw that there was still SMC6 positive cells, and this was about the same proportion of cells that we had at the end of treatment. And then we had, of course, also some other cells where we saw that this transcription was ramped up and they were full of HBV RNA. So we believe that the rebound we observed in general was due to the rebound in these cells that hadn't managed to degrade, to bring back SMC56 and silence cccDNA. And on this slide, I would like to speculate a bit about the possible mechanism of the sustained silencing and possible reactivation. And with these staining pictures, I also like to recapitulate what I showed you before. So there's active transcription without treatment of all viral RNAs, and then when we treat with siRNA, we see that SMC56 comes back and silences transcription from cccDNA. However, when we stop, there's a viral rebound. When we shield the hepatocytes from new infections, we see that cccDNA silencing can be maintained. So then, of course, the next question is, how does this reactivation happen? And apparently NTCP-mediated infection events of either complete virions or even virus-derived particles appear required to either introduce active cccDNA molecules, new ones, or reactivate the suppressed ones. In the setting, we cannot differentiate between these two possibilities, but there was a recent study that just came out showing that inactive cccDNA molecules can actually get reactivated by superinfection. And they showed this in this elegant study where they superinfected HBV core deficient virus in cells that had been infected with HBV-X deficient virus. And then they could see the reactivation of the cccDNA by a core staining. Then the next question is, how does HBX do that? How does HBX get into the cells to reactivate or even to establish normal cccDNA transcription? And this can either happen through the uptake of HBV mRNA containing particles, because it was shown that in serum there's also HBX mRNA, or it could also happen very early from incoming active cccDNA molecules. So to sum up, the primary mode of RNAi is to degrade all HBV RNAs and inhibit viral replication and protein production. And the secondary mode of action of RNAi is the cccDNA transcriptional silencing through the reduction of HBX and the reappearance of SMC56. And the silencing also seemed to be long-lasting, but new infection events appeared involved in the reactivation and cccDNA rebound. So there are still many questions to be answered, and all this data I showed you was in preclinical models because we don't have any data, as far as I know, in patients about this mechanism of action and how this relates to the treatment outcome we see. And we don't even really know if SMC56 even plays a key role in patients in HBV activity. We don't know the overall contribution of this potential effect in treated patients. And if we have this effect, we also don't know if cccDNA suppression can be maintained after treatment. And of course, this is a very relevant question for patient treatments, because if this is the case, then we should treat patients with doing a rebound with agents that can either lower viremia, NUCs, or actively inhibit new infections. We also don't understand the fate of silent cccDNA molecules. Do they just hang around waiting to get reactivated, or are they eventually degraded? And all this data I showed you in the mice was in a model which is immunodeficient, so we couldn't analyze any adaptive immune responses. So the next important question would be if this overall effect is sufficient to restore HBV immune responses. And with this question, I would like to hand over in a second to the next speaker, to Matteo, but not before I show you at least one snippet of data from patients. So this is a liver biopsy of one highly viremic chronic HBV patient, a stain for core and SMC6 again, and we actually see a very similar pattern to what we see in mice. So there's SMC6 in the uninfected cells, but in all the infected cells, the cells that are positive for core, this factor is absent. So I believe this restriction and this interplay really plays a big role also in patients. And the next burning question then is, of course, what happens in patients after RNAi therapy? So with this, I'd like to acknowledge the people who worked on the mouse studies that I showed you, namely Katja, Mark, Tassilo, Moritz, Johannes, Rudy, and Simon. And I thank you for your attention and happy to take questions.
Thank you. Thank you very much, Lena. Open for a discussion. If you would like to make questions. Over there. Sorry, I can see you at.
Yeah, that's right. Great talk, Lena. I just wanted to ask about that final bit of data. Was that an e-antigen positive patient?
Actually, I'm afraid I don't have a lot of data on this patient because it was an old archived material, and I have to go to the paper archive to retrieve this information, but I will. And I'm also hoping I will get my hands on more patient biopsies.
Yeah, definitely.
And so if you happen to have frozen biopsies, I would be very happy to collaborate and do more stainings of this kind.
Yeah, that would be great, because I'm just wondering whether these foci would be... Okay, it was core staining, is that right?
Yes, it was core staining, so it was actively infected.
The question is some of that X coming from integrations, and do you see any that are negative or both?
Yes, of course, that's of course the next question. I'm trying to perform other stainings to find out.
Beauty, thanks.
Luca.
Going back to that thing, please remember that when you see nuclear core staining, that doesn't mean that the cells is actively replicating the virus. It just means that it once encountered the virus, but nuclear core staining can stay for weeks, if not months, in a cell that has been cleared. So that doesn't mean that those cells are replicating the virus.
Okay, thank you for your remark. I will perform also RNA in situ hybridization to actually check if there's active replication. Thank you.
Okay, I think there are two critical points. The first one is X promoter is unique and might be weak. And secondly, so we have to understand, so X has no structure available yet. If I put like C-terminal, I put full length X into AlphaFold, I could see a clear possible C-terminal structure, but N-terminal is very flexible. So that may actually inhibit the drug development. So, but my question is actually, do you think it's possible, or have you compared actually the expression of HBx in vitro versus in vivo? Because in vitro, actually, what we have found, so the phenotype of Y type and X minus at the same OI, for example, in HBG2 and TCP, the phenotype is the minimum. It's larger in HyperG and PHH, but if you look at the transcriptional level, it's only like 5 to 15-fold in my hands at least. But I think in vivo, as far as I understand, the window is larger, so can you comment on that?
Actually, I haven't done a head-to-head comparison of HBx levels in vivo and in vitro. I'm not sure if in this publication, the Korneyev et al. Or the de Corsia paper, they've done this. But unfortunately, the antibody is, it's always a problem with the antibody. We have to do a pull down and then a Western blot to detect it. So the levels are not really abundant, but yeah.
Stefan.
Very nice overview and very nice talk. I just want to make a comment on the amount or the grade of the transcriptional silencing if you compare an X minus virus versus an X positive virus. I don't think, or we have no evidence that it is complete transcriptional silencing. It is at the max 100-fold silencing, but an X minus virus can replicate, make cccDNA in the same way, but it is not that it's completely shut down. You can have a cccDNA from duck without any X, and you get transcription. Maybe in the duck, this is overcome by the copy numbers that are just amplified and not under regulation of X, but it's not a complete silencing. It is just a damping of a factor of maybe a log or 2 log.
Yeah, I know we've had this discussion before, and I think in vitro it's true with the X minus, but in our vivo experiments with the RNA in situ staining, we can clearly see that there's not a single mRNA coming from these SMARC positive nuclei. So I think in these cells that have SMC56 coming back, we don't see any transcription at all. And this, the staining is very sensitive. But of course, we have to keep this in mind when we compare in vitro data and in vivo data, yes.
And maybe also on that, I mean, in the original paper in Nature, they also use plasmids. It's not something that's extremely specific for cccDNA. It is an episomal regulatory mechanism that X can influence. That would mean that you can completely silence the plasmid as well. I don't think it's complete silencing.
Okay.
But perhaps, Lena, a little bit following this and still looking at primary human hepatocytes and in some in vivo situation, what I think is still intriguing, yes, there are cells that are completely silenced, but I think the targeting of the liver, and this is a really important question, why a decent amount of cells still show in the nucleus small dot of RNA expression. We don't know exactly which RNA was that. And I think this is important because this is the source for the rebound. And this, to address, I think, this question is important. I don't know, do you have an idea why?
Yes, that's an important point. Maybe, well, maybe the treatment wasn't long enough, or maybe just some cells don't make it, don't manage it. But I don't know why, because in the staining we see that in the cytoplasm all RNAs are degraded. So eventually HBX should go down and then SMC6 should come back, but it doesn't happen in all the cells. Maybe it also doesn't happen in all patients then.
Last question?
Yeah, well, I think this is actually sort of relevant to the question you just asked, and Thomas was asking, right? Those few cells still expressing something. Could that be derived from integrated DNA?
Well, in our, maybe, just know that in our mouse model, there are integrations, but they are very little. And I don't think these integrations could account to this many cells still producing RNA.
In the chronic infected patients, that's certainly possible, right?
Yes.
The many integrants.
Yeah.
Okay, I would say we close now. Thank you very much, Lena.